All right, good afternoon, and thank you for joining us for the first Red Talks online. Rensselaer responds to COVID-19 research resources and resolve. This afternoon, we have more than 400 registered for this exciting presentation. This is the first of a series of Red Talks online, RED standing for Research, Education, and Discovery. These webinars will be led by Rensselaer researchers who highlight and discuss our approaches and solutions to global challenges. A few housekeeping items before we begin. Please make sure your volume is turned all the way up. You will be muted throughout the presentation out of respect to our presenters. You will see that there is a Q&A feature at the bottom of your screen. Please feel free to type in your questions and we will get to them as time allows during the Q&A portion of the program. I'm now pleased to introduce our host and moderator for today's discussion, Dr. Robert Hall, Acting Vice President for Research, Henry Burlage, Jr. Professor of Engineering, and the Director of the Center for Materials, Devices, and Integrated Systems at Rensselaer Polytechnic Institute.
Well, good morning, everybody. So good afternoon, rather, I'm sorry. Welcome to the first online Red Talk, Rensselaer Research, Education, and Discovery presentation. Our Red Talks epitomize research in the new Polytechnic and how it integrates with the education of our undergraduate and graduate students. So Tom has been good enough to introduce me. My name again briefly is Robert Hull. I'm our Acting Vice President for Research at Rensselaer. I want to thank you all for joining us. In particular, we're honored to have the 18th President of Rensselaer, Dr. Shirley Ann Jackson, on this event, and at least some of our trustees, including John Bennett, Arthur Gajasa, Nancy Muller, Kate Murta, and Curtis R. Preem. And if there are other trustees on the call, again, thank you for joining us. So by the numbers, the research enterprise at RPI comprises over 30 research centers, around 1,000 graduate students, countless undergraduate researchers, and over $100 million in annual expenditures. It is organized into 5 signature thrusts according to the Rensselaer plan. The first is biotechnology and life sciences, where Rensselaer researchers address the molecular mechanism of disease and help find cures. The second is energy, environment, and smart systems, where Rensselaer researchers are developing new energy resources and are mitigating the effects of energy consumption on the environment. The third is nanotechnology and advanced materials, which provides the fuel for new technologies and new solutions to global challenges. The fourth is computation, data, and artificial intelligence, which underpins research across all fields of knowledge and virtually every technology. Finally, the Media, Arts, Science, and Technology Thrust is epitomized by our Curtis R. Preem Experimental Media and Performing Arts Center, or MPAC, where scientists, artists, and engineers come together to create new knowledge and new forms of expression. So these signature thrusts align with the most pressing global challenges facing humankind, such as human health, the environment, global warming, future energy resources, stewardship of critical resources such as food, water, fuels, and scarce elements, and physical and cyber security. To address these challenges, research at Rensselaer integrates the frontiers of engineering, basic sciences, computation and data science, and cognitive and social sciences. This intellectual marriage provides innovative solutions to such global challenges, including mitigating disease, providing clean food and water, developing new sources of clean and renewable energy, and establishing a resilient national and international infrastructure. We are inventing new methods for advanced manufacturing that lead to new products and processes. We are developing new materials for advanced computing, energy storage, and medicine. We are pioneering new technologies to manage the explosion of data from new communication networks and control systems. And most urgently right now, we are addressing the greatest health challenge facing humankind for a century, the COVID-19 pandemic. So Rensselaer researchers from all 5 schools, engineering, humanities and social sciences, arts and social sciences, science, the Lally School of Management and the School of Architecture are at the forefront of the fight to mitigate the effects of the COVID virus and ultimately to defeat it. The efforts of Rensselaer researchers have received national headlines across a range of endeavors, and a select number of those are shown on this slide. In particular, AMOS at RPI, which is the 24th most powerful computer in the world, has been purposed as part of the COVID-19 High Performance Computing Consortium it's been purposed to computational work to address the challenge of COVID-19. And this was actually announced live on national television by the President of the United States. So now in this presentation, we're going to now see a particular example of the work of Rensselaer researchers before we move on to our panelists. So this is one where Rensselaer engineers, scientists, and biologists came together to address a critical and pressing need in the fight against COVID-19. We're working with the Mount Sinai Hospital system in New York City. We're addressing an urgent need for a immediately usable sterilization system to allow reuse of N95 masks for those at the very front lines of the battle against coronavirus. So I'm going to pass back to our technical expert here, Tom, to run the video, which just for a minute and a half or so.
We received a request from the senior leadership at Mount Sinai describing us that there is a situation and the situation was getting worse with the shortage of the personal protective equipment. Various hospitals were trying to look at ways of repurposing the PPEs. So could we bring in the creative expertise of Rensselaer? This really was an interdisciplinary group which formed, and everybody wanted to contribute something.
This quickly transformed into a proposal to build a mask disinfection system that we could build quickly and hopefully automate in a way that people would be able to use to disinfect thousands of masks a day. We convened, started assembling very quickly using standard available assembly parts, then started actually measuring UV dose required for what we thought was going to be the dose necessary to disinfect masks. UVC radiation is a line of sight kind of dose. If there's any shadow from the mask or any kind of thing blocking the access of the UVC radiation to the mask, you're not going to disinfect that part. We elected to come up with a system that allowed us to run the masks vertically so we could simultaneously dose the masks from both the front of the mask and the back of the mask. And this is somewhat unique.
These are anxious moments for all of us looking at what's going on in the country and also across the world. And we all wanted to, do something to make a difference. And I think this is an example where we came together to deliver a solution. And hopefully this is going to be useful.
So now I'm going to introduce our 2 panelists for the discussion to focus on the biological and biomedical research at Rensselaer that is addressing this coronavirus challenge head-on. So the first of our panelists is Dr. Deepak Vashtish, who is the director of the Center for Biotechnology and Interdisciplinary Studies at Rensselaer. He has been with Rensselaer for 20 years. He joined as an assistant professor in 1999 and then rose through the ranks to become the department head of biomedical engineering and is now the director of CBIS. As director of CBIS, he has expanded interdisciplinary research and our key partnership with the Icahn School of Medicine at Mount Sinai to address global healthcare challenges in Alzheimer's, cancer, diabetes, precision and regenerative medicine, and most recently, COVID-19. His areas of expertise are biomolecular science and engineering of the extracellular matrix with applications to aging, osteoporosis, diabetes, cancer, and Alzheimer's. Dr. Jonathan Dordick is the Howard P. Eisenman 42 Professor of Chemical and Biological Engineering at RPI with joint appointments in the departments of biomedical engineering and biological sciences. He joined Rensselaer in 1998 as chair of chemical and biological engineering. He has also served as the previous vice president for research and previous director of the Center for Biotechnology and Interdisciplinary Studies, and is currently special advisor to the president for strategic initiatives and co-director of the Heparin Applied Research Center. Dr. Dordick has made foundational contributions in biomolecular discovery and engineering to address clinical translation in the areas of infectious disease, including COVID-19, neurological diseases, and anticoagulant therapy. Both of these panelists are internationally recognized leaders in their fields, so it is my honor to moderate this discussion with them. So now we're going to have a half-hour discussion with our eminent panelists, and then we'll be taking questions from the audience. So I'm going to start with a question for you, John. And it's a very basic question, but one we all care about deeply. What is happening with this virus? How long will it last, and how many people will it infect?
Well, thank you, Robert, and good afternoon, everyone. Yes, it's kind of the most basic question, what's going on and what's going to happen with this. And of course, you have to keep in mind that just a couple months ago, we didn't know a whole lot about it. So we're learning so much more because of the research around the world. It's obviously very transmissible because nobody's been infected with it before. As a result, up to 3 people would be infected for everyone who has the virus and transmits to those people. But we're bringing that down because of social distancing and among other things, and that in many areas has gone below 1, which means the actual numbers are dropping, and that's quite important. We don't have immunity to this, which means that the virus will continue to spread unless we do social distancing or we come up with various ways to abate that through various therapies, or until, of course, we end up with a vaccine, which would be effectively being able to provide that immunity to everyone who would be given that vaccine. It's insidious in the sense that there are a very large number of asymptomatic people who are pre-symptomatic. In fact, the virus spreads under both those conditions. And one of the things that we're learning a lot about is that viral load plays a major role. That's why cities that are very dense, like New York City, for example, have so many infections, whereas other areas of the country will have much fewer infections and maybe even fewer severe infections because the amount of virus that a person's exposed to would obviously drop. So these are just some of the things that are underway. Obviously, more people will be infected, and until we get to the point where we have a vaccine, that's going to be a concern.
So thank you very much, John. So follow-up questions for you, Deepak. Will it come back after this initial spread, this spring and hopefully not much longer than summer? Will it come back? Can people get reinfected? How fast and how much will it mutate? And what are the most serious comorbidities?
Thank you, Robert, and good afternoon, everyone. Robert, in terms of the questions you asked about the mutation rate, so clearly, like all viruses, this virus will mutate. The good news is that it does not mutate as fast as some of the other viruses. And it has, in fact, it's 2 to 4 times slower than the common flu virus. And that leads to the idea that as we talk about traits like virulence and how it may affect, those traits are regulated by multiple genes, and that's unlikely to happen. So in terms of mutation, yes, but it's unlikely to become more virulent and affect many people in such a short time. So we're talking about a short time, 6 to 8 months here. So second wave, as we've all sort of read, is predicted. But the idea is that as we go towards this particular phase, we would have some kind of immunity, because clearly all kinds of exposures will give you some degree of immunity. And animal studies have shown that the exposure does help increase the immunity to the virus. So that's sort of the good news moving forward.
So thank you very much. So now, looking forward, what are the timelines for an effective vaccine? We hear from the Jenner Institute at Oxford University that they hope to have a vaccine available by as soon as September. How realistic is this? How quickly would a vaccine be effective in eliminating the virus, particularly if the virus continues to mutate? Will we always be chasing the cure, as we are, for example, with the flu? And finally, what is Rensselaer doing in this realm of finding a vaccine? So, John, I was going to ask you to answer first.
Yeah, so, vaccine timelines are quite long. They're among the longest in any kind of drug development. And part of that, of course, is that in the end, you're giving a vaccine to a healthy patient. It's not a treatment, it's a preventative. And so that makes it more complex. But you're right. I mean, Robert, you're an alum of Oxford, so of course you got that in, which was very important. But they had a head start. They actually were developing vaccines against a predecessor, the MERS, the Middle Eastern Respiratory Syndrome coronavirus, which is another, in fact, quite a bit more dangerous coronavirus, although it didn't spread as much. So they were already partway there. They were developing at least parts of the necessary machinery in which case a vaccine can be developed. So, everybody's waiting for something like this. There are many, many different routes today that people are using to generate a new vaccine. Some of these are very novel. So the hope is that there will be a vaccine generated in the next maybe 3 to 4 months that would actually go. There's already a few that are going to patients now in a phase 1 trial, but some that would be in a much larger number of patients in a trial. A key point, though, is how do you scale that up? It's one thing if you're going to have a vaccine for a fairly small number of people. When you're going to be needing to inoculate perhaps in a large fraction of the world, that's going to take a lot of scale up. And that's a major component of things that we are doing here at Rensselaer. So, and the last thing I would say about this is that even though the virus may mutate a bit, and it probably will under a number of conditions and over time, an imperfect vaccine is still far better than no vaccine. You're still going to get significant immunity. We know that with influenza, for example, we don't often hit the perfect vaccine in a given year, but it actually provides still a significant immunogenicity that you have to protect you from getting a very severe illness.
Thank you very much, John. And Deepak, what would you add on this particular point?
So, Robert, I want to highlight, not in the context of vaccine, but some of the work which Stephen Kramer, one of our faculty in chemical and biological engineering, is doing. So he's working with Johns Hopkins on the National Convalescent Plasma Project, in which they are extracting plasma from patients. And Steve and his group is working to scale up the production of neutralizing antibodies. So that could be provided as a treatment and for prophylactic treatment also. And it allows you to have greater antibodies available for testing protocols as well. So I think this is sort of running in parallel to our efforts in vaccine, which can be helpful in the short duration.
And thank you. And that actually translates very nicely into the next question, which is, what about therapeutic approaches? Repurposing existing drugs or designing new ones? What is Rensselaer doing in that realm? And if we can ask you to lead off on that one, Deepak.
Sure, yeah. So I'll give you 2 examples, Robert. One is the work of Guy Monteleone, who's one of our new Constellation professor in the biotech. So he found that if you study the structural aspect of hepatitis C virus protease, and one of the proteases, he evaluated it, which has a very big similarity to SARS coronavirus 2, and in terms of its proteases, so they have a similar mechanism of action. So that leads to the fact that some of the drugs which have been used for hepatitis C could then be repurposed for addressing COVID-19. So he's done some fundamental computational studies, and we're looking at a variety of drugs which can be deployed. And a second example is the work of Chinyu Wang, who is a faculty in our Department of Biological Sciences. And Chinyu is looking at the various compounds, and he's looking at a library of more than 30 million compounds. And you mentioned, how Rensselaer combines all these interdisciplinary approaches, looking at computational with structural biology, and scaling of all these compounds and synthesizing some of them as a potential therapeutic. This is not a short-term solution, but eventually it'll take some time before we can, make these come up as a therapeutic level. But that's an interesting and a new approach to looking at COVID-19.
So thank you very much. I'll pass over to you, John. I know this is an area particularly near and dear to your heart. We're doing very well on timing, so please do feel free to take 5 or 6 minutes to the brilliant ideas you and your colleagues have in this area.
Okay, and actually there's a show and tell here, so if you want to put back a few of the slides, I'll give everyone a sense of 2 different areas that are underway. One of them we call viral traps, and this was actually developed and led by my senior research scientist, Seok-Jun Kwon, and actually his son was the first author on this paper, and his son got his BS at Johns Hopkins after 3 years, and he came back here to Rensselaer to do a bit of work before heading on, as well as Professor Xin Wang, who's moved recently to the University of Illinois, Bob Linhart, who's our colleague and the Biocatalysis Metabolic Engineering Constellation Chair and a faculty member in chemistry and chemical biology, and myself, really an interdisciplinary approach where we actually developed something that involves what's called DNA origami. It's basically a DNA that you can manipulate into a variety of different structures, in this case, a star-type structure. And this was developed for dengue virus initially, but also moved on to Zika and some influenza. And we're modifying this exact type of structure of the DNA. It's really a synthetic DNA in this case, so that it actually fits the structure, the fingerprint of the COVID, of the SARS-CoV-2 virus. So we can tailor it in any number of ways. And that's important because when that trap gets literally onto the virus and it traps that virus, the virus will end up lysing or disintegrating and, of course, dying. And so that's one of the major traps or goals that we have, to kill viruses like dengue, but we also assume that it's going to play a significant role, or could play a role, in other viruses as we move down the road, including this coronavirus, maybe others as well. The other thing, which probably would actually be used even earlier than a therapeutic, would be a detection system. And so we can use the exact same approach to be used to detect a virus with tremendous fidelity. And so that allows us to effectively have a test strip, very much like a pregnancy test strip that one can use. In this case, you could take saliva, for example, and then be able to determine whether or not the virus, in this case, the coronavirus, the SARS-CoV-2, exists. You would be able to see that within minutes. And so it's really a point of care home testing system, but it can be tailorable to other viruses. And you can actually look at having a test strip like this that would be done at the same time as a separate strip to determine whether or not you have influenza. So this is a very direct way and a very quick way of being able to detect whether or not we have a virus, or a patient would have a virus. And that is, of course, at the front line. We're going to talk a little bit more about detection systems soon. Another approach that we've taken actually involves something that is near and dear to my heart, which is anticoagulant drug heparin. If we take a look at this next slide, we developed a multifaceted therapeutic approach with heparin. It actually started before we learned a lot about the coronavirus, but we knew enough about the coronavirus to understand some of the molecular structure. In particular, how does the virus actually infect a human? We know that the spike protein, which are spikes on you see in the corona of the virus surface, binds to the ACE2 protein, the ACE2 receptor in the human cells, such as in the airways, in the lung. And it turns out that heparin, which is the world's most common anticoagulant drug, binds exceptionally tightly to the spike protein. In fact, it binds in 3 separate sites, essentially wrapping the spike protein up within the polysaccharide that's heparin. The binding is exceptional. It's in the picomolar range. Really, that means it's even stronger than an antibody. And that's quite amazing. In discussing this interaction between heparin and the spike protein, we were in contact with colleagues at Mount Sinai, and we found that they were starting to see something rather unusual. They found that a number of patients that were in serious condition, those both in the ICU, maybe even some on ventilator support, were unfortunately obviously getting the ARDS, acute respiratory distress syndrome, but it was different than a typical ARDS. In fact, what they and other medical centers around the world were finding was that the lungs were more compliant. It was more like something along the lines of a pulmonary embolism. And so heparin began to get used along with other anticoagulants in a protocol that Mount Sinai and now others are using as a current therapeutic pathway. And I'll just mention one other thing. Heparin's been known for now 100 years. It's been known for a long time to be anti-inflammatory. That's not how it's used, but it turns out that the course of infections like the COVID indicate that really it's not the initial viral infection that does the most severe damage to the person. It's likely the cytokine storm or the inflammatory response. And so heparin plays a major role there. So those are just some of the examples that we've taken underway to deal with the therapeutic opportunities of a drug, common drug like heparin.
All right, thank you very much. So, so the next question is going to focus on testing. And I'll ask the question, and John, while you're on stage, we'll ask you to lead off on this. So how critical will testing be? And I know the short answer is very, but how available and accurate are tests now and say 3 months from now? And what is Rensselaer doing in this realm?
Yeah, well, it's impossible to overstate the importance here. Everything we're talking about, everything we at the university talk about, everything that the federal, state governments talk about is, do we have enough tests? We need the tests so we know who's infected and who's not. We need tests to know who has been infected and therefore may be immune at least for a long enough period of time to really get back into society. So there are 2 very specific types of tests. One involves the actual live virus. That's the one that you have people that have the swabs that go up their nose. Appears now that we might even be able to use saliva, which would make things a lot easier. And that involves basically the molecular biology tool, the PCR, the polymerase chain reaction, which people may know of because that's actually used in forensics to determine whose DNA was it that may have committed a crime. But it's the same kind of approach. So it measures basically, in this particular case, with very high fidelity, the RNA that the virus has as its genetic machinery. The problem, of course, is do you get a good enough sample? That's where the false negatives come in. The antibody test is becoming increasingly important. That's measuring, do you have an antibody in your blood that indicates that you were exposed to and have had the disease. And there, that's really picking up significantly. And within the next couple months, I think we're going to have a test that's going to be a typical blood finger prick type of test, which we have now. But there are too many false positives, but that's getting resolved. And I think within a couple months, we're going to have an extremely sensitive and accurate test for that. The combination of this needs to be ramped up dramatically. That is right now the biggest challenge. How do we ramp up to over 1 billion tests for the virus? That's what we're going to need, and probably 500 million or 300 million to 500 million of the antibody. That's going to be the biggest challenge that we face.
So I was hearing on the radio this morning about a massive supply of tests, I think from China to India, where the false positive, actually the false negative rate, was 50%, which is about as bad as you can get, I guess, right? I mean,
yeah, it means essentially the test means nothing.
Nothing, yeah.
We're not nearly that. I mean, the live virus test, when the sample is taken correctly, is essentially 100% accurate. And the antibody test, right now, there are some that are close to 99% accurate. For an individual, that's perfect, really. For a population model, that's still going to be a challenge because you have a large number of non-infected people, which means that they magnify dramatically the false positive effect.
Yeah. So, Deepak, what are your thoughts in this area?
A lot of things are coming up. So one interesting example comes from our partners in New York, Mount Sinai. So you've probably seen in the press that they recently got a approval for an antibody-based testing for COVID-19. And it's a very interesting approach they applied that, you know, that the coronavirus uses its spike protein to latch on with the ACE2 inhibitors in the lungs. And the issue is that if once you have that, the immune system cells will respond their own defense and will produce antibodies against these spike proteins. So the way they designed this test that they take spike protein and then look at the blood samples from the patients who've been exposed. And if there are antibodies against spike protein, the blood literally lights up from that point. And it's a pretty, straightforward approach. There are issues with antibody design, but that's something they have got the FDA clearance of, and they are going to be using it, and they've in fact started using it. So I think we can expect more, like in our region and other places, more players like this coming up and coming up with tests.
Thank you. Thank you. So far we've concentrated on the near and intermediate term future. So this last and final question is focusing on the here and now. So what is Rensselaer doing now to address the immediate needs of those on the front line with personal protective equipment? Deepak, maybe you could lead with that answer, then I'll contribute something to this particular discussion.
Sure. Thank you, John. So I think that's a very important problem, and it's a problem at hand. And as you showed in the video at the beginning of our Red Talk, that Mount Sinai and also other local hospitals have contacted Rensselaer that, given this crisis of shortage of personal protective equipment and 95 masks in particular, face shields and others, what can Rensselaer do to help them into that situation? And this is a great example, and I felt very humiliated, very humble about this particular aspect, that when this problem was put to our faculty, they all came together. And it's, again, one of the new example of the new Polytechnic model that we had LISA, which is Lightning Enabled System and Application Lab at RPI, headed by Bob Karlichek, working with a civil engineer who designed a conveyor belt system. And then coming on to chemical and biological engineering, looking at the viral load testing, we were quickly able to develop a system to disinfect N95 masks. And we built up the system in a record 2 weeks' time, shipped it over to New York, where it's currently being evaluated to see the killing efficiency of the source against SARS coronavirus 2. And we're looking forward to this result with great expectations. So that's one approach which uses the current PPEs. One of the other projects comes out of Department of Material Science and also the Department of Chemical Engineering by Ed Palmero and Helen Zha. And what they have developed is they have done a polyelectrolyte thin film which can be sprayed on to provide, extending the use of it and improve the barrier function of the mask, so they can be, neutralizing the coronavirus and extending the life cycle again of these PPEs. So I think these are some of the examples which, we are working on and several others around the country are working on to provide an immediate solution to people at the front lines. And you have, I'll pass it on to you to share something else.
Yeah, so that's just one other example in this, addressing immediate needs arena of PPE. So Albany Medical Center approached us probably a few weeks ago now about, at that time, their greatest current need was face shields, essentially to protect the mask to allow for longer use and wear. And they came to us with a crowdsourced 3D printed design. And in short order, we put all our 3D printers to use, and we generated our first shipment of about 400 shields to AMC quite recently. But because we have great engineers, we have great staff, we have great faculty, we have great students participating remotely. We were able to come up in just a week or so with a much improved process using plastic injection molding for the main frame of the mask. That is something which will speed up the assembly and creation of these masks from probably 2 hours each to about 2 or 3 minutes each. So we can be thinking about the thousands. We've had great collaboration with local companies such as Industrial Tool and Die, and great ingenuity as well. We're talking about the key materials limitation for these things are the plastic visor material. You can't buy it for love or money because everybody's trying to do the same thing. And one of our team, actually, Paul Caccione from this company I mentioned, Industrial Tool and Die, found an untapped source, which was these sort of thick plastic document binders you use to put together documents. And that was about the one untapped supply we found, and probably Staples and Home Office very confused as to why there's this massive run on this particular item. But I love that sense of materials ingenuity and engineering solution there. All right, so this has been a great discussion. I'm going to ask both of our panelists to spend a minute or two at the most just to address sort of concluding thoughts on what the greatest challenges and what the best hopes are that face us in the next year or even years. So John, I'll ask you to start on this, if I may.
Yeah, I think I have 3 points really in terms of what's going to be needed. In the short term, we need the testing. We need to get the testing that's going to be accurate, it's going to be sensitive, and it's going to be in bulk. And it's most likely going to need to be local. So local communities will have to have testing that is going to be just as effective across the country as you're going to have in your local community. The second thing is we're going to need to have a therapeutic intervention. There's some small molecules that may show hope, certainly neutralizing antibodies that we've talked a little bit about, as well as potentially large-scale production of antibodies that come from the convalescent plasma. That's over 100-year-old technology, and it actually works. But you can't scale it to the level that we need, and that's what we'll need. And finally, of course, we need a vaccine. And maybe I'll say the last thing, which is we need the U.S., the people, the government, and particularly government here in the U.S. as well as around the world, to understand that infectious disease is really important. And we are seeing that now. So we need the research community to be supported to drive infectious disease-related work, which will obviously benefit everyone.
So thank you very much, John. So Deepak, your concluding thoughts, please.
Thank you, Robert. And I do agree with the points John put forward. I think those are significant challenges. To add to the list, I think one of the immediate need I see is we should be looking more carefully at drug repurposing. So this is the solution which may be available to us at hand, maybe not as a cure-all, but something we can rely on to address the problems we are facing. And John alluded to some of those examples, whether it's heparin or some of the other drugs we are looking at. And I think we need to look at more carefully at this particular aspect. There has to be that need put forward. And this requires a systems approach. So I think we are also looking this problem in isolation. There are issues of comorbidity, for example. We know that diabetes, hypertension, and older age groups are more susceptible to this crisis. So what is so typical about these diseases and what we can learn from them, so that we apply a systematic approach. So drug repurposing, a systematic approach. And the third thing, I think it's a challenge for both the biotechnology and the material science community, that how do we scale up productions, whether it's of crucial drugs, whether it's vaccine, antibody production. So finding them is one thing, but then scaling up the production, given the pandemic, is going to be really important. And the same applies to PPE. We talked about the personal protective equipment. How do we make PPE in a new and different ways that it is more liable, more conducible to sterilization, for example, in the long term. So we may have to look at them fundamentally differently as this crisis goes behind us.
So thank you both very much for an absolutely fascinating discussion. So what we're going to do now is turn it over to the audience for questions and answers. So the first question I have here, and I'm going to try and just make this so I can read more of it, is there any way RPI can suggest Dr. Glanville distributed bio to develop his antigen treatment? So I would say please follow up with an email with us on that. You can email me at, type this in, hullr2@rpi.edu. All right, so that was from David DeWitt. Thank you very much for that question, but it sounds like there's a more detailed thing we want to get into that. How hard would it be to refit the oxygen delivery system in airplanes to deliver clean, sanitized, coronavirus-free air supply? Can something be done? So bear with me, I'm just trying to go back and see that question. So can I ask, Deepak, would you take the first attempt at that, or first answer to that? How hard would it be to make the oxygen delivery system airplanes deliver clean, sanitized, corona-free air supply? Can something similar be done in subways or with a person on the Captain Connect? I would love to know more about that because I recently went to Tibet and learned firsthand the powers of supplemental oxygen, so we say, because of the altitude there. So Deepak, will you take the first shot at that question?
Yeah, I mean, there are a couple approaches one can think of. We've looked at HEPA filter, for example, that how efficient the HEPA filters are in taking on the coronavirus and other particular viruses which pass through the system, and there are ways of modifying it. We talked about sterilization and also putting the polyelectrolyte solution we talked about with the work of Helen Zai and Ed. Some of those approaches may be put in to modify and retrofit the existing system. A lot of it depends upon the testing which we are doing, that how effective are these approaches against the coronavirus too, and if they appear to work, then we should definitely look at these questions which have been brought up by Michelle in that particular area.
So there's a question from our provost, Prabhat Jailer, asking about why there is such unreliability in current tests. And John, you touched on that earlier. Would you answer that question or amplify on that question?
Yeah, so there's different answers based on a different type of test. So on the live virus test, the problem in terms of reliability comes from being able to take an appropriate sample. Because once you actually do the physical assay, the real assay that's done with the RT-PCR, it's extremely sensitive and it's very, very precise. In fact, it's about as close to being 100% accurate as you can get. So, but the problem is you get enough of a live virus in the sampling. And since that's the human element that comes in, I think that's where you're seeing a large number of problems. There are some new tests now that don't have to go all the way back into your nasopharyngeal region. You can take a little bit easier test or recovery of the virus. And in some cases now there's some talk, and there's been some initial FDA clinical approval on this, in simply taking saliva after coughing a few times and taking a saliva sample and then being able to do that. That would be ideal because it's easier to deal with. And I think that's going to dramatically reduce the false negatives. In terms of the antibody test, the false positives, that, of course, really depends on how good the antibodies are that you're going to be able to get. Again, this has only been relatively recent, so they're getting better and better. There's been a report out of the University of California, San Francisco, showing which tests actually work well. And some of them are hitting up to the 98, 99% true positives. And so I think we're going to see that get to the level that we need for both individual analysis, individual care, as well as, of course, epidemiological modeling.
Thank you. Thank you. And we have a question for one of our trustees, John Bennett, which I think I'll direct to you, John, again, which is any update on the heparin study?
Yeah, so that's actually underway. There are a number of hospitals that are now looking at broadly anticoagulant therapy, including heparin, both low molecular weight subcutaneous heparin that would be in the general hospital setting, as well as potentially the unfractionated heparin IV that would be used under more ICU conditions. Of course, you have to worry about bleeding, which is a common concern with an anticoagulant. But there's been decent evidence thus far to indicate that it is playing a significant role as part of a group of anticoagulants, as we've learned a lot more that this disease is more of a vascular disease than it even is of a pulmonary nature. So it may start as a respiratory infection, but then likely the impact of the immune system and the cytokine storms leading to significant vascular problems.
Thank you very much. So there's a question from a name I recognize here, Hank McElroy, and asking about data analytics work. And we will be having an entire new Red Talk on that at some point in the near future. And I think that will be discussed at the end of this presentation. So it's a great question, Harry. It's going to have its own moment in the sun with people like Jim Hendler and John Kolb and Chris Carruthers within a few weeks.
But I would say, Robert, that yes, that's going to be a huge part of how we're going to address this problem. And we're on the front lines of that. So yeah, that's very important to stay tuned.
That's a really good point. That's a really good point. So, and we will be, as I say, bringing you the latest and greatest on that in a separate Red Talk. So a question from Carlos Garcia, a question that either Deepak perhaps first could answer. A group in Spain reports success with the use of bovine lactoferrin plus vitamin C. We claim 100% recovery in infected patients. So what are your thoughts on that? Deepak and then John.
Can I have John go first? I'm looking at the question. I didn't get that question fully, Robert. So let me.
Right. Yeah. So John, did you catch
Yeah, I mean, so the proteins like lactoferrin actually are used to transport things into cells. And so being able to transport perhaps a highly redox, antioxidant species like vitamin C get into cells may help a bit. There are a lot of various approaches used that are not very dissimilar to this. It's very hard to know really how many people truly have been enhanced, the treatment's been enhanced by this without doing your classic double-blind clinical study, or even if it's not double-blind, still having the necessary control groups. I think that's still something that needs to be done with many of these. Some of them are being done like this. And I wouldn't doubt that there's some examples along those lines that will be somewhat protective. Again, what you really want to do is do something at home so you don't go to the hospital. If you do go to the hospital, you want to do whatever you can to keep them from getting into the ICU. And by the way, simple things at the ICU may prevent people from going onto a ventilator, such as turning them upside down, putting them on their stomach. It actually is preventing a number of them from progressing to the ventilator.
Thank you. So actually, I think I'll give you a different question, Deepak, which is, as we look at repurposing drugs, is there any hope for hydrochloroquine?
It's a very interesting drug. Clearly we've heard enough about it on the national media. I think with hydrochloroquine, I think there has to be a controlled trial, and a lot of them are going on, and we need to look at the results. But I think the question is that in some of the countries like Brazil and India, which have a lot more experience because of malaria with this drug, the idea is to investigate it being taken up as a prophylactic one, and at an earlier stages to see that before the whole COVID-19 process sets in, what are the impact at the very early stages. A lot of time it is being tried where patients are coming to the hospitals who are already at an advanced stage needing a ventilator, and perhaps that's not the right time for it. But clearly, there are side effects, as we know, with cardiac and other issues related to that. So it has to be evaluated in a very careful manner, and it may not be the right medicine for everyone. It may work in certain cases well. And I think I'll leave it at that.
So there's another great question about essentially epidemiological modeling. That was from Tyler Housel. Is RPI doing modeling to determine best practices based on different approaches with local and national governments? And again, great question. And again, that will be one of the major focuses of our upcoming Red Talks, our very next Red Talk, in fact. And there's a few really great questions about, what does the new normal look like on a campus, on research? When might students be able to return to campuses? And they're great questions. I just think, we probably would have a different group of body to answer those, different body of people to answer those. I would just say we think about this each and every day. So maybe that can be an upcoming discussion. Here, I think we're going to focus more on the biological aspects of COVID, because these questions we could be debating until this time tomorrow and still not run out of thoughts and ideas. So thank you for being with me and sending that questions. So another question is, while you're on deck, Deepak, are there very early signs of COVID-19 before the fever of cough? Lower oxygen levels or higher blood pressure or breathing rate? Are there even earlier warning signs that might be indicative before the fever is too advanced?
I think we've all heard about the loss of smell and loss of taste. And I think that's an important one. Clearly, the low oxygen levels have gained a lot of attention, which is, a little bit after the onset of fever, or it can be at the same time. And other common things are like sore throat, common flu function. So I personally think that it has to be a combination of factors. If you can combine these factors and come up with a score, which will tell you a little bit more objectively how to move forward. And this is, again, where data analytics is going to be very helpful. That looking at these normal one thing off at a time may not be the right approach. And that's clearly not working.
So here's another question I'll pass to you, Deepak, from another great friend of Rensselaer. I know everybody, that applies to everybody on this call, but John Capac. Given Rensselaer's work on UV sterilization of PPE, is there a dose which can be applied to surfaces which would also be safe for human exposure?
That's a great question. And hi, John, how are you? I think there is a great example from one of our alums, Colleen Costello, who started this company, Vital Vial. And that company specifically looks at the effect of ultraviolet light on surface decontamination. And they use the ultraviolet light in the visible range, which is 400 to 420 nanometers. And it leads to photoactivation of molecules inside the microbes, leading to cellular damage at the end. So they already have a product which they have been promoting, and that came out of Rensselaer, and that is something which we'll all get much more interested in, and probably potential users as time go by. So not UVC, but as John correctly points out, looking at ultraviolet light in the visible range would be much safer to apply for that kind of decontamination.
Thank you very much. So there's a question from Tom Eisenbud, which I will address to you, John, asking about, will there be a test soon which will not only detect active disease, and antibodies, so both active disease or antibodies? So you either know you have it or have had it. So John, what's your response to that?
Well, the tests exist. It's just a question of getting them out there and being able to have them be as accurate as possible. So the antibody test, which is the one that will tell you whether or not you have had the disease, and if it's sufficiently past the point where you don't show any further symptoms, and quite frankly, a lot of people don't have any symptoms, that test exists, and I fully expect within about a month to 6 weeks, not only will it become even more available, but I think it's going to have much greater accuracy, much fewer false positives, which will be very, very beneficial for everyone who would take that. I'll sign me up to do it. I'd love to be able to take one of those soon. And then again, on the live virus test, that's really a throughput issue. And the ability to take a good sample and have a high throughput, and to do it, if you haven't had the disease, you're going to have to be tested every so often, maybe every couple weeks. That's why it's really a question of ramping that up to excessively large scale.
So I think, though, John, the question was, is there any test which will do both?
Well, you could, yes. They're both based on well, they use two very different methods, but you can even do a live virus test in a manner similar to that of an antibody test. Actually not looking at the genetic material, the RNA of the virus, you can actually look at the virus itself. And so similar to that approach that I had shown with that test strip, there are a number of approaches now that are being developed to do even live virus tests on a test strip. So that might be perfect if you stick it in your mouth, for example, and get a bit of the saliva and then run it, just like a typical type of test strip that you can use, like a pH strip, so to speak. So I think that's something that's going to be coming. I know there are a number of companies working on that, for example, with CRISPR technology. So I imagine that's something that will be available sometime this summer, at least in testing.
So there's a question from Elizabeth Peck. If an infected person is asymptomatic, how long are the shedding the virus of how long is one potentially infectious with others? And what is the mechanism by which an infected person is so asymptomatic? I mean, how can they basically avoid all symptoms? So Deepak, can I ask you to answer that?
Sure. So obviously, you know, a lot of people have been asymptomatic as they're infected, and it depends on the level of exposure, and secondly, their own immune system, that how quickly the immune system reacts to it, and are they having any comorbidities, for example. So if there are comorbidities like diabetes, they're already low inflammatory situation in these individuals. And there are other aspects of our gut microbiome, how we all have different microbiomes. So we have different levels of low or high grade inflammation, and those factors may predispose some people. We know that older individuals and certain races and ethnicity and people with some pre-existing conditions are more. So I think all these things stack up together in terms of determining a person's response and how they will react to an exposure. And maybe, John, you want to add something to it?
I think you hit on the big ones. I mean, except for the fact that, quite frankly, this is a major question we just don't have the answer to yet. And boy, would it be great to understand why some people remain asymptomatic. And I think Deepak hit on the major reasons for that. And I think the immune system is going to be in the middle of all of that, of course, including how rapid the immune system ramps up to attack the virus and then maybe goes into overdrive. And I think that's where we run into the symptoms that one sees.
So, John, while you're on deck, we have a question from Jim Hodler, who is the director of our Institute for Data Exploration and Applications, will be one of the stars of the next Red Talks. So he's asking basically about how soon do we get to home tests for COVID? He makes the analogy to home pregnancy tests. How soon would you say we are from that being realistic?
So I think it's going to be, well, let me state that some of the examples already exist. So for example, these test strips, one can do similar to a glucose strip, because you have glucose for diabetes, you have to have a little bit of blood, a pinprick, and be able to do that. That could be very easily done at home. And you're dealing with your own blood and you're putting it onto a little test strip, and by capillary action, simply the blood on in this case, the antibodies that you may have will flow down the strip and it'll either light up or it won't. And that's something that's being developed now and in fact is used now. It'll be improved and we can do that. The live virus test can only be done that way, similar to the answer I gave previously, if we have a strip that does not require PCR technology, so that you can measure the actual full virus, not just the RNA, and not an antibody response to the virus, which is what the blood test does. That's going to take a little bit more time, but again, I think that there are approaches out there that we're going to see at least described in detail probably within the next 2 months.
So thank you very much. We have a question from our Dean of Science, Kurt Brenneman. And again, I'll keep that start with you, Kurt, and then line up the next couple of questions with John, rather than the next couple of questions for Deepak. So Kurt is asking about the question of reinfections, and are they largely attributable to earlier false negatives?
I believe the answer is yes. I think that the expectation among physicians, immunologists, and people studying this is once you have been exposed, once you've generated antibodies, there's and given the linkage of this with other respiratory infections, as well as other coronavirus infections, such as some that had to do with the common cold, the expectation is you're going to have immunity for some period of time. The real question is, how long is that time? Is it a year? Is it 2 years? Is it reduced immunity but still protective? We don't know that yet. I think what you've seen in these reinfections is almost surely the fact that it was a false negative on the virus test, and perhaps because very little is actually being shed. And so you go from false negative, and then the next test you show up positive. That doesn't mean you were reinfected. It just means you didn't pick it up that first time.
So there's a question here that says that infants are sometimes born with COVID-19 transferred through serum or plasma. So Deepak, does that happen?
I am not familiar or aware of this particular report being out. Clearly, there is a connection because of between the mother and the infants, prior to birth, but I'm not sure whether these will be passed on.
Yeah, John, do you have a thought on that? Because that's a very sinister, scary thought.
Yeah, I don't know, and I don't want to speculate on anything. I think that's obviously something we have to be concerned about in terms of transmission from a sick mother to the baby.
Yeah. So we've got so many great questions here, and we still have well over 200 participants. So we will go for just 5 more minutes and trying to get through the majority of questions, not all, I'm afraid. There's a question about any use for nanotechnology here. There's somebody very active in the field of nanotechnology. I'm going to claim that molecular level engineering like this is all nanotechnology. So when John and Deepak wear other hats, they might call themselves nanotechnologists very comfortably. So the next question is about the effect of the microbiome on the severity of COVID-19 illness. Deepak, I think it was you who referred to that. Can you amplify further? I think I just got a text from Deepak that he has connection issues. So John, I'm going to pass the question.
We've been finding out so much about the microbiome in the last few years that my general answer is yes, there's going to be a relationship there, like with everything else. So I truly believe that the microbiome plays a major role in the immune system, and therefore it's going to play a major role in how people respond to the coronavirus and, quite frankly, every virus and bacterial infection. And so I would imagine it's going to be, as I said, there's going to be some significant linkages that we're going to find out, including how we can prevent the severe infections from any virus.
So thank you. So the next question I'm going to pass on to Deepak initially is to do with ventilators. Are there improvements considered for ventilators? And really, this is Spiro Marazakis, who is pointing out that a small percentage of those getting on one ultimately manage to be cured, and we know that. And what is the reason for that? I think, for example, Boris Johnson, the Prime Minister of the UK, is one exception to that unhappy rule, I believe.
He didn't go on a ventilator, though.
That's right, he went to the ICU, not the ventilator, yeah. So it's a great question. Deepak, will you take the first pass of that? Why do people not get cured on ventilators? Why does it seem, unfortunately, too often to be a one-way sort of vector? Can you answer again then, John? And we'll see if Deepak gets back.
I think keep in mind that we're looking at, and well, I'll be direct on the ventilator part. I mean, we're looking at the sickest of the sick that go onto the ventilator. So a large number of them likely are so sick going in that it's very difficult. And of course, what you're doing is stressing the body on that. So I think that that creates a concern. The other thing to keep in mind is we're really dealing with the tip of the iceberg. And, you know, the vast majority of people are either asymptomatic or have very mild disease. Small numbers go to the hospital, and of those, a smaller number go to the ICU and onto the ventilator. And so when you're dealing with that cohort, it's very difficult to know what commonalities exist about why some people do go to the ventilator, why some don't, why some on a ventilator are able to come off, and maybe the vast majority do not come off. I know our governor has said basically the longer time you stay on a ventilator, the worse the outcome. And I think that's clearly the case that we're seeing here.
It's about the time he wrapped up anyway. Again, I will give you my email, and if you've got something, you can write it down, or you've got a great memory, and perhaps this will make sure this goes out with the follow-up from this presentation. But it is HULLR2 at rpi.edu, and I can certainly direct traffic at this end. And I'm going to ask Tom now if you can come on and show us the last slide, which is kind of your wrap up. And when you've done that, Tom, if you can let me get back on and just say thanks to our audience and just a couple of final thoughts. So after you, if you want to go ahead, Tom.
Thank you, gentlemen. Thank you to all of today's panelists for their time, and to all the attendees here today. As Dr. Hall stated, a fund has recently been developed that will support research at Rensselaer. Please visit giving.rpi.edu for more information about this fund and other ways you can support Rensselaer. We hope you enjoyed today's presentation. You will be receiving a follow-up email with a link to today's recording, along with a survey. We would ask that you take a few moments to complete. Please stay tuned for more announcements on future Red Talks online webinars. Please stay safe and enjoy the rest of your afternoon.
Thanks very much, everybody.
Thank you all. Thank you, John. Thank you, Deepak. Thank you to all the participants, all the attendees. Thank you.